Hood, we'll get back to aging and Alzheimer's and the connection, but first, a curious thing, because you talked about using worms. And the first thing, well, do worms get Alzheimer's? Why do you use worms? What's the benefit?
Oh, it's a very good question. But worms are a very well established biological research system nowadays. And there are several reasons. First, when you want to study aging, you'll better have a short-lived animal if you don't want to age before your subjects.
You get your results.
Yeah, exactly. And worms, on average, they live only for 19 days. I'm talking about small nematodes called C. elegans.
Except if you give them heat, they live for 60 days.
No, give them heat. You can extend it a little bit, but by stressing them, but it's a different issue. These worms have several advantages. Some of them scientific and some others are practical. So I mentioned the practical issue of having short lifespan, but scientifically, we can manipulate their aging process very, very easily using technique which is called RNA interference. And by just feeding them bacteria, and that's their food, they eat bacteria, which express some genetic constructs, we can manipulate the aging process. It's much, much easier than manipulating aging of mammals. This is one thing. Another thing is that these animals have very simple pathways. So unlike mammals, the insulin IGF signaling pathway in worms is only one. It's a merged pathway. And the downstream components are much simpler. Instead of having 4, we have only 1. So it's a great system for searching, for screening, and for studying the very basic fundamental mechanisms. However, it has to be said that at a certain level, you need to validate the findings and you need to test the relevance for mammals. And that's what I actually have done. I worked in worms first, reached a certain level of knowledge which can be achieved in worms, and then moved on to the mice.
To validate what you found in the worms.
I would say to validate and elaborate on, because after finding the specific pathways, we wanted to test first that it works the same way in mice. But mice enables more comprehensive research. For instance, if we're talking about Alzheimer's disease, it's very important to test the cognitive functions such as memory, orientation, which are very difficult or maybe even impossible to test in the worm. But there are well-established assays for memory impairments and cognitive functions in the mouse. So we validated and elaborated on the mouse.
Okay, now when I read a few of your papers, and we'll talk about the specific papers a little later, I formed an image, and I want to know if it's accurate. The image was, the real problem is this garbage collectors are on strike. They're not picking up all these leftover proteins and they're piling up. Or else there's no compactor to reduce them in size. That's the image I formed. It wasn't, I don't blame you for that. Is that any accuracy to that?
I would say that in general this is correct, and maybe I'll say a few words about the mechanistic problem which leads to the development of these disorders. Okay. So cells encounter challenges from all types constantly. One of these challenges is protein aggregation. And when protein is produced, it has to undergo processing in order to become functional and to attain three-dimensional structure and to actually mature properly. However, a certain level of, or certain fraction of newly synthesized protein fail to attain this desired three-dimensional structure and expose hydrophobic domains. Now, hydrophobic domains you can, I would say, describe as sticky domains. It's very similar to the little oil that you have on your soup. And you know that those little drops, they don't like to stay apart. They want to create one large drop. That's exactly what proteins do. And we call it protein aggregation. Now, in some cases, protein aggregation leads to disease. Now, what size of droplets are the most toxic? So it has been suggested by others and validated by our studies and others as well, that small, what we call oligomers, small aggregates are most toxic. So cells develop two kinds or two methods in order to get rid of those toxic species. One is to disaggregate them and degrade them, which is the best way to solve the problem because practically it's gone. However, when this pathway is overtaxed or it doesn't function very well, there is a second line of defense, which is compaction or higher aggregation.
I see. So it's sequential. You do.
So we believe that this is sequential and that the first priority, the preferred pathway, is to recycle the amino acids, to degrade the bad proteins, which would solve the problem permanently. However, if it doesn't work well, and we believe that the aging program, the genetic aging program, actually mediate decline in this activity, there is a second line of defense.
It's a backup defense.
Yeah, what we call the compactor. So, in general, that's correct.
I'm glad. I got good marks. But you also have the reason for that. You talk about chaperones. What are chaperones and how does it relate to this mechanism?
So chaperones are proteins which assist other proteins to fold properly and take care of those who did not fold properly. These are actually sets of proteins which serve as, we call it sometimes housekeeping genes or proteins which are responsible for the integrity of other proteins.
Well, I had an image there too of telling your kids, you see, if a protein does it properly, you've got to do the same job when you fold your clothes. And put them away.
That's correct.
It's a similar operation.
It's very similar. But of course, they have several roles, not only to assist proteins to fold properly, but also take care of those who did not fold properly. And if their activity is impaired or negatively regulated by aging, problem is around the corner.
I see. So that's now how does that connect up then with Alzheimer's?
So Alzheimer's is one disease in a group of neurodegenerative disorders that collectively, and actually there are several others which are not neurodegenerative disorders, called
Parkinson's?
conformational diseases. Parkinson's and Huntington's are part of the same group. Now in Alzheimer's, there is a small peptide called A-beta, and A-beta is very aggregative and it tends to form aggregates. Now when these aggregates accumulate, they cause damage to certain substructures in the brain, a process which eventually leads to disease, to the onset of Alzheimer's disease.
But as I read it, it's not, this is the plaque that's formed, is that the plaque? Or
so plaques are actually places where fibrils, which are high molecular weight aggregates, accumulate in an extracellular localization. However, it's not necessarily, in other diseases, it's not necessarily an extracellular localization. Some other diseases, it's in intracellular. There's a whole variety of deposition sites of large aggregates. However, it was shown for several diseases that the most toxic species are the oligomers, are the small and not the large aggregates.
I see. So that's similar to your basic research, in the same that you have to get just a proper amount. You can't have it too little or you can't have it too great.
Yeah, that's correct. And it's apparent from our work. It's suggested and shown by others as well in different aspects and different disorders.
And what I want to do is explore this a bit further, not from the science point of view, the basic science, but how it gets applied to diagnosis and treatment. But we're going to take a break and then come back to that issue if you can stay with us. Don't leave.